Hello guys, today we will start with the other types of hypersensitivity. We have already discussed the hypersensitivity type 1 and today is the day for 2, 3 and 4. Hypersensitivity 2, it has also got some other names, that is antibody mediated and cytotoxic. We will discuss why these names are given to the hypersensitivity type 2. When it comes to hypersensitivity type 2, we should remember two things, that is, these are tissue specific and preformed antibody are present. What does that mean? The tissue specific, that is, for example, if we take the thyroid, then the antibodies are related only to the thyroid tissue. They won't go to any other organs. The preformed antibody, that means, as we saw in hypersensitivity type 1, the antibodies were formed after the first contact, but here the antibodies are already present. So as soon as the antigens enter, they will start acting with the antibody. So now what happens when the antigens enter? The antigens combine with the antibody, which triggers the complement system, and which stimulates the formation of the macrophages, lymphocytes. What is the combined name of antigen and antibody? Yes, it's of course called the antigen antibody complex, but apart from that, it has got another name, that is immunocomplex. And what is the complement system? It is the group of proteins which stimulates the formation of the macrophage and lymphocytes. Then these macrophage and lymphocytes, what do? They go to remove the immunocomplex. They remove the immunocomplex. In this way, as the whole process, it was the antibody was already present and the immunocomplex was formed with the addition of the antibody. That's why it is called the antibody mediated hypersensitivity 2. Now, for example, if we take, there are examples like autoimmune diseases, Graves' disease, and the most important example, that is the transfusion mismatch. If there is a transfusion mismatch, then it is the hypersensitivity type 2. How? As in hypersensitivity type 2, the antibodies are already preformed. Suppose a person with blood group O has antigen beta and he has been given with blood group B. It is just an imaginary example. So these will kill the blood. So that's why it is called that the antibodies are specific to the RBC. If there is a mismatch, then the antibodies will react. That's why it is the hypersensitivity type 2. Then comes the hypersensitivity, I will write it as HS type 3. What happens in HS type 3? Here again, we need to know that it is also antibody mediated. So again, here will what happen? The antigen will come, antigen plus antibody, then together they will form the immune complex. Then in the same way, these will trigger the formation of the complement system, that is the group of proteins. But here what happens is the complement system does not stimulate the formation of the macrophage. So there is no formation of the macrophage. So there cannot be, as there was formation of macrophage in the hypersensitivity 2, so there was remove of the immunocomplex because of formation of macrophage, but in this there is no formation of macrophage, so the anti-immunocomplex cannot be removed. So what is the defect here? The defect is the failure of the immune system to remove immune complex. As the immune system was unable to, the complement system was unable to form the macrophage, so the immune system was unable to remove the immune complex. That's why this hypersensitivity has also got another name, that is immune complex reaction. Now we will go further for the examples of this. These are mainly found in the joints, that is the arthritis or rheumatic arthritis. We can find these in rheumatic arthritis, hypersensitivity type 3. Then in small blood vessels, so they will be called as vasculitis. And in kidney, in the form of glomerulonephritis. So this is the difference between type 2 and type 3. There is formation of the macrophage and lymphocytes. Here there is no formation of the macrophage and the lymphocytes. So because of the formation here, it is able to remove the immune complex, but as there is no formation of the macrophage, it is unable to remove the immune complex. So there is failure of the immune system to remove the immune complex, which leads to the hypersensitivity type 3. Now let's go further to the hypersensitivity type 4, HS4. These, as we have already discussed, the hypersensitivity 1, 2, 3 are immediate, but hypersensitivity type 4 is delayed. It is the only hypersensitivity type which is delayed. It has also got the name as T cell mediated and cellular cytotoxicity. From cellular cytotoxicity, I got to remember we also discussed about cytotoxicity here, but we did not discuss about it. This I will discuss after I tell you about the hypersensitivity type 4. So this is completely mediated by the T cells. The T cells are again divided into CD8 cells, which is also called the killer cells, and CD4 cells, which is also called the helper cells. So what happens here is, when the antigen comes, it goes to the APC. What is APC? The antigen presenting cells. Then from here, the APC, it goes to the CD8 or the killer cells. Then through the lymph node, it goes to the CD4. Then the CD4 leads to the macrophages. And again the same thing, this macrophage will remove this antigen. As this was the case in the hypersensitivity type 2. But here we notice something, that all of these are cells. The antigen presenting cells, the CD8 cells, the CD4 cells, the macrophage cells. and that's why it got the name as T cell mediated, as both the T cells, the CD8 and CD4, are responsible. Now it got another name, that is the cellular cytotoxicity. Cellular is the cell, cytotoxicity is the damaging. So as this is completely damaged with the help of all the cells only, that's why it is called the cellular. But in hypersensitivity type 2, it got cytotoxic name because antibody was involved. Antibody was already present. There was no name of any cell. Therefore, it was only cytotoxic reaction. And here, as these all are the cells, it is called cellular cytotoxic reaction. Now, when we go for some examples, the most common example is the tuberculin test. This tuberculin test, which has protein mycobacteria, this is injected into the skin of the person. This is the skin. This is injected to the skin. What happens is, if the person has been exposed to these kind of tests earlier, then there will be dermatitis. If the person has been already exposed, and there will be thickening and hard induration of the skin, hard and thick skin. This will happen in the tuberculin test if the person has already been exposed to this tuberculin test earlier, not for the first time. And the tuberculin test has also got name as Montauk's test. It's based on the scientist name. The again second example is the contact sensitivity. Contact sensitivity. It's very understandable from the term, the contact of anything. It may be we are allergic to so many things, the leaves or something. When they come in contact with our skin, it again the same thing, the skin gets inflamed, the dermatitis go, and hard and thickening of the skin. But apart from the contact sensitivity and tuberculin test, there are some systemic diseases which also leads to hypersensitivity type 4. Systemic diseases like rheumatoid arthritis in the joints. Next is the multiple sclerosis will be there. Multiple sclerosis, that will be the myelin around the nerve fibers. So it will be around the nerve fibers. And next, it will be lining of intestine in the bowel diseases. So hypersensitivity type 4 is different from the rest type of, because it is cell mediated while the others are antibody mediated. It has got the T cell mediated name, it is the delayed one, and it is the cellular cytotoxicity reaction. So this was about all the three types of hypersensitivity, their examples with their mechanism. I hope you understood it. Thank you.